My name is Cora, and I'm going to outline the structure of DNA as described by Watson and Crick and how the structure gives DNA stability. Watson and Crick's model of DNA, known as bDNA, with 10 base pairs per turn, is right-handed. Coils can be sorted into left and right-handed based on which direction they turn to. For example, the plastic coil is left-handed while the shoestring is right-handed, like most DNA is. The way DNA coils in the double helix creates major and minor grooves. These alternate throughout the structure to allow for larger or smaller sites for proteins to bind to. DNA is composed of nitrogenous bases that pair and have a sugar phosphate backbone to form a double helix. The nitrogenous bases are grouped based on their structure where purines, adenine and guanine, have two rings, while pyridamines, thymine and cytosine only have one. Bases pair specifically so that in normal conditions, T binds only with A and G only with C. When pairing, thymine and adenine have two hydrogen bonds and guanine and cytosine have three. Hydrogen bonds are relatively weak as hydrogens can only have one bond and an attraction to another base. Therefore, H bonds provide bond specificity rather than structure. Instead, base stacking caused by hydrophobia stabilizes the structure. Bases are mostly nonpolar and are therefore hydrophobic or water avoidant. To maximize their avoidance of water, the bases will stack upon each other to create minimal surface area, which keeps DNA in a twisted double helix structure. To make the double helix, the nitrogen bases connect to sugars, known as nucleotides, that are able to utilize phosphate to create the sugar-phosphate backbone. A nucleotide is a 5-carbon ring completed by an oxygen. The primary carbon connects to the nitrogenous base. The second connects to a hydrogen in DNA or a hydroxide in RNA, hence why DNA is a deoxy-RNA, as it has one less oxygen than RNA. The third carbon will bind to a phosphate that is then connected to the fifth carbon in order to create phosphodiester bonds, which have negative charge. Due to the specificity of the third and the fifth carbons binding to phosphate, and because DNA is antiparallel, one strand will be 5' to 3' and the other will be 3' to 5'. The phosphodiester bonds present will give DNA an overall negative charge. This is a destabilizing force as one negative strand will attempt to push the other away, but stabilizing forces are able to overcome and keep strands together. We can measure the effect of negatively charged strands by placing DNA in a high concentration salt solution, such as saline sodium citrate buffer, which will partially neutralize the negative charges. Then, we can quantify how stable DNA is by measuring the midpoint of denaturization, T of M, where half of DNA has been separated into single strands. With less negative strands, stabilizing forces such as base stacking will be more effective, and it will take longer for DNA to separate into single strands. In addition, strands of DNA will become less hydrophilic or water-loving and more hydrophobic as they lose their negative charge. Thank you for listening.